Good evening, everyone. I hope I am audible. And in the last class, we were discussing about gene library using EMBL3A. I mean, the goal was to first understand, am I audible? Somebody confirm, please, before I move ahead.
Yes, sir.
Okay, good. So we have been using the protocol that you're viewing right now for to explain at least two things. One, if it is about phage vectors, and the second one is about genomic library or gene library. So I think in these two ways, you have the best understanding of it. And if you still have any questions, you can always ask. And are there any particular questions or any topics that you need to repeat for CA1? You need me to repeat, please let me know ahead. And you can WhatsApp so that I can plan up and discuss in that way. So tomorrow we don't have a class, right? Okay, day after tomorrow is the exam. Well, so in today's class, we are going to go ahead with trying to understand a few more vectors. So we have discussed about plasmids, we have discussed about phage vectors, or at least lambda EMBL3A vector, which is one of the complex ones and a very smart one. And if you understood that, you understood majority of the phage based vectors. And here is a cosmid. Cosmid is, say, when we were reading about plasmid based vectors, we discussed about one of the best advantage is they are very simple and they are very stable. That is a good advantage they have. But then there is a limitation. Limitation is, the serious limitation is size of the fragment that can be cloned. And why is that? Why is the size a limitation? Size of the fragment a limitation? Typically, we don't really have to, theoretically, at least plasmids have everything that needs to be, that needs for stable propagation. They have an origin of replication. We would have constructed one with a selectable marker, right? An antibiotic resistance gene. Then how is the fragment size being limited? We also discussed this before. The point is that when we have made a recombinant, say if this is a recombinant plasmid with a fragment of interest, this is a fragment, and then we need to put it into bacterial cell host. The frequency or the probability with which they can be transformed into the bacterium is a serious limitation, because the larger the size, the lesser the frequency. So about, say, a total size of 10 kb is fine. Kind of, it's still less in probability that the number of transformants one would get, but still it is possible. But if you want to clone beyond that, so something like in phage vectors, in the previous one we discussed, is we were able to clone up to 20 kb fragment. We cannot do that same with plasmids because the limitation is with packaging, is with transforming the host cell. What if we could circumvent that? What if we could somehow put it into the bacterial cell? And that is where people have exploited coscyte. So they have taken a plasmid, like an example like this, PJB8, which has an origin of replication, beta lactamase marker, and then there is a cloning site, BAMH1, unique cloning site. Now they have put coscyte. Why did we put coscyte to start with? I hope you remember the significance of the cos site. Cos site is important for in vitro packaging. The idea is simple. As I said, the size is the limitation, right? The limitation is the size. So we want to put somehow into the host, and why not manipulate or exploit the ability of phages to carry the DNA of our interest into the cell, right? Then it's like, as we saw in the previous one, in vitro packaging, and it has a kind of active system of pumping in the DNA from outside into the cell. So if I want to use such phage-based injection system, right, then I should be able to pack the DNA that I'm looking at. The DNA should be packaged into the phage particles. How would the DNA be recognized and then be packaged into the host, into the head particles? That's the question. If we remember in the last class, when I was discussing about the phage biology, lambda phage biology, we discussed that from theta mode of replication, it forms rolling circle, resulting in concatemers of lambda phage like this. Each one is a lambda phage. It's something like a stretch of each joined end to end. That is how they are produced, concatemer. And each of these, what I have indicated, is a cos site. So the distance between this is one lambda phage. On either side, you have a cos site like that. So the head particles recognize the cos site and then pump the DNA inside until another cos site is experienced. In concatemers, that is how they do it. So if I want to package any DNA into the head particles, this is specifically lambda phage, then the DNA should have a cos site. Only then it will be packaged. So they took a plasmid and put a cos site in there. Okay, and now you can, there is a possibility you can theoretically put in somewhere around 20 kb fragment or 10 or 15 kb fragment and still be able to package in by using in vitro packaging into head particles. And that means you're artificially assembling the phage particles, but they are actually hybrid. The genome of it is the cosmid that we put in, right? It's not lambda phage. So we make these phages and then we infect the host cells, and these very efficiently will put the DNA into it. And then we have a circular, very large fragment, a large plasmid, plasmid with the large fragment of interest into the cell. And that is the reason why cosmids were made, and cos site was specifically included into it. Okay, that is the advantage of using cos site. The key is everything about how can we put the DNA into the cells. Okay, those are cosmids. Cos, and this one has come from plasmid, cos, and mid, cosmid. So we are going to also see a few more. Here is something called M13 phage. It is important for single strand vectors. It has interesting biology. It is unlike lambda phage is a double stranded DNA, and M13 phage DNA is single stranded, and it is plus. I hope you understand what is meant by plus and a minus. It is a filamentous bacteria. It is, phage, filamentous bacteriophage, which is enveloped. So theoretically, it can grow any size. All it needs is the proteins, more number of proteins, and it can form something like this. We can also have, there are phages and other are like this. The head particles, if you see, they have a strict structural limitations, or they cannot have larger size because the number of capsids they fit in is perfect. It should be proper, right? Because otherwise it cannot make this exact shape. And if there is space constraint because of the restriction on the size and shape, you will also have restriction on the amount of DNA you can actually put in there. Whereas something like 13 phage, M13 phage has a structure like this. It's not restricted. It is, you can have as long as possible in this one. All it needs is some proteins on one end and some other proteins on the other end. It infects bacteria that are F plus, I think, which has pili, and they infect and they enter into the cell. And they have single-stranded DNA, as is shown here. And then once it is once it infects a host bacterium, the second complementary strand is synthesized, which is called as the minus strand. So here we say plus strand because that is what is found in the M13 phage. Okay, normally when you say plus strand, all the mRNA is a plus strand because it has an open reading frame that can produce a protein. The minus strand would be complementary to the plus strand. But here, I think this is given as plus or minus based on what is present in M13 phage. Anyway, and then once it's infects the second, it is converted into double-stranded DNA, and it is called as replicative form. And this replicative form can undergo several rounds of replication, amplifying the number of present, because you have one single-stranded DNA, and it is now converted into one double-stranded DNA, which is then converted into multiple single-stranded DNA. Okay, and at one point, when it is supposed to produce more phage particles, it will synthesize single plus strands, and these will form, along with the other proteins that are involved, these will form the phage particles, and they are released out of the cell without lysing the cell. Not that everything has to go this way, but M13 phage will keep on. It's like M13 phages are budding out of the cell, right? It's not necessarily like a lytic cycle. So I hope you can see the good things about it are, the good things are, this is about 5 kb or so. Okay. Say, for example, you have a bacterium, and the bacterium is infected with the single-stranded, the M13 phage. Inside the cell, it becomes double-stranded. Now, what you can do is isolate it and manipulate it as similar to that of a plasmid. I mean, you would have used a plasmid kit to isolate the DNA. You can just isolate the DNA, do all the manipulation as if it is very similar, simple, small, double-stranded circular DNA. You can then make a recombinant DNA. You can put the gene of interest in it, and then allow transformation of it back into a different host. And now you have the recombinant plasmid, right? And if you wait, eventually it will form some phages and give out single-stranded DNA like this, as in the form of phages, M13 phages. So it is a very attractive one because it is a phage, but you can play around as if it is a kind of plasmid because it's not very big. And theoretically, it is limitless how much amount of DNA you can actually clone. But of course, it's not practical in many situations because it gives single-stranded DNA. But there are many instances when you're learning about gene manipulation or molecular techniques and so on, where you have to produce single-stranded DNA, especially something like using for sequencing. If you want sequencing, which we will learn again in Sanger's method, or mostly it is about Maximum Gilbert method, we will learn it. Although it is obsolete, we will still learn because the logic is good. So in those instances, we need single-stranded DNA. And it's not possible for us to take a double-stranded DNA and try to make it a single strand and get it. Again, I'm not saying that it is impossible. I'll say one strand as A and another strand as B. And you have, say, for example, 10 double-stranded DNA molecules. If you have heated it and you have separated them into 2 test tubes, what is the guarantee that all A's will be in one tube and all B will be in another tube? That's not how it be. There will be some A's and some B's like this, and if the temperature is less, they will again reanneal and form double-stranded DNA. So if you want only the single-stranded DNA to form, this is a very good way of doing it. Also, when we do southern blotting or not southern blotting, especially because here we are producing single-stranded DNA, we need to make probes. Now the probes can be chemically synthesized and all, or there are different ways. But in some time ago, they used to make probes using this. Probe should be a single-stranded DNA, and then this is a very good one. There is also one more that's not listed here. You will also get to do site-directed mutagenesis, which we will all these things are some things that will come in the future in our course. If you want to do site-directed mutagenesis, then there are in some protocols you need a single-stranded DNA. So these are M13 is a good and very comfortable vector for generating single-stranded DNA. Okay. Now we'll move on to a little more others, which you know it as expression vectors. I think we already had an introduction to it. The one of it, first thing is what we learned is cloning vectors, where the objective is just to increase the number of this, say, gene X by putting into bacterium and then multiplying and isolating. Eventually, what you're doing is you're increasing the number of copies of X. That means you're cloning X. When you want to know what exactly the gene X does, what does it code for? Is it RNA or is it protein? And what does that protein do? Then you need to express the genes. And if you want to express any gene, we need to have a promoter, right? So, and there is one more thing that we need to remember is every time an open reading frame is indicated as an arrow. This side, it would be the start codon, and on the other side, you have the stop codon, TGA, TAA, or TAG. So the arrowhead indicates the direction of the open reading frame, right? If it is an RNA, it is a different issue. And the promoter should always be on upstream of the open reading frame. This would have been the downstream of it. This is the upstream. So all those vectors that have a cloning site downstream of a promoter would have been indicated this way. Those are, and which have the possibility of expression. So for expression, we need several things, especially if it is an open reading frame. You need, first, transcription should happen to produce mRNA, and then translation should have to produce the protein. So if we are cloning only the open reading frame, and we have only the transcription, then we will get the mRNA, right? And for the mRNA to be translated and to produce a protein, we need the ribosome binding site. Anybody, what is the ribosome binding site in prokaryotes? Anyone? What's that? Are you there with me?
The shine diagram sequence.
That's right. It's called as Shine-Dalgarno sequence. And here would be the start codon, and this is the stop codon. In eukaryotes, it's called as the Kozak sequence. Eukaryotic translational initiation is little different, of course. Okay? So the ribosome binding site should also be there. So first thing, expression vectors, especially if you're trying to express a protein, a gene to form a protein, then we need a promoter and a ribosome binding site as well. Okay, all those can be called as expression vectors. And you also have a term here, transcriptional fusion. In the next one, we will see translational fusion. If there is a gene, an open reading frame from a subject host that you're looking at or you're studying, trying to study, and it doesn't have its own promoter. There are two ways of cloning also. Suppose you have a cloning vector, and it has the gene that you have, gene X, and it has its own promoter. You can clone from here, amplify from here to here, and clone all of it. So you have a promoter, which is its native promoter, and this is the gene of interest. And it could be transcribed and probably translated also to produce a protein, and we can study that. But often, if you're working with eukaryotes, and our host organism is majority of the cases, it's E. coli. And you cannot express those, the promoters don't work in E. coli. E. coli transcriptional machinery recognizes only E. coli type of promoters or related ones. So what we can, in that situation, what we can do is we should then be having a expression vector which has promoter that can be recognized by the transcriptional machinery of the E. coli. And here is gene X, and then probably you should also ensure the ribosome binding site, and only then we will be able to get large quantities of this protein, and that protein can be further characterized for its function, sequence, and other things. Okay. Here, the gene of interest is from eukaryote, and we are putting in a vector, the promoter from a promoter from E. coli. We are fusing two different things. Those can be termed as transcriptional fusions. There is actually, the best way you can probably see is you can have a bacterial gene here like this. Okay. And you could probably also clone a eukaryotic gene here. And these will be co-transcribed, isn't it? In prokaryotes, the mRNA are polycistronic. That means they are present as operons. One promoter might be involved in expression of more than one genes, like lac operon, for example. You have LAC-Z, LAC-Y, and LAC-A together under the promoter of lac promoter, and they will be produced, transcribed equally, at a time, co-transcribed, and each of them will be translated by ribosome to give rise to three different proteins like this. So this is also called as this can also be the one that I'm saying, this is of bacterial, and assume this is gene X from a different organism, and it will produce mRNA, something like this, and eventually it will give rise to protein B and protein X. That is also transcriptional fusion. Okay. Then we're going to see what is a translational fusion. Translational fusion is, this is a promoter, and assume that I have a gene of interest which has its start codon, and here would have been the stop codon. Okay, I'll put it this way. If I am cloning a gene, in continuation with this. That means the stop codon is no longer there. This would have been the start codon, and the stop codon would have been replaced. And here would be the stop codon. Now, if the transcription occurred, mRNA would be produced like this, and translation for the ribosome binding site is here, and the ribosome would start translating from here, and then will go on translating until the stop codon is here. So it will then produce a different type of protein. It will produce a fused protein, right? Half part of it is of that of the, say, bacterial, and the other one is that of X. So because we are fusing the open reading frames, this is into one open reading frame. If you remember, if you can see here, these are two independent open reading frames. Whereas in this case, this is one single open reading frame. That means the cloning site should have been exactly here. And the reading frame should have been in continuously without any frame shift mutations or any alterations. So if one is trying to do this, then one should have been very careful reading through the information so that when it is produced, when the protein is produced, it is this fusion of exactly these two proteins. If we add or miss one or more of the bases here, we might have a shift in the reading frame here, and the protein that will come here, the sequence would be something else, right? So that is one thing people should take care of it. So these are called as translational fusions. They have their own advantages, and we will discuss them at the later time. Okay. So with translational fusion, I want to take you with I will take you with some applications today, and that will be the end of today's class. Here is some of the proteins, say especially if they are very small proteins. Okay, if they are very small proteins, or some proteins are really toxic. I'll just get into some theory. Why do we need fusion proteins? If translational fusions are possible and you can get the protein by itself. Because if we get a protein like this, it's not, if we cannot study the function of it directly, because half part of the protein does not belong to it. If there is a function, and then we don't know which part of it is actually doing that. So if we want to study the protein, it should be like that, just made of sequence what should have been for X, protein X. But sometimes it is difficult because they are small or they could be toxic. Say, for example, I take a cell and I have a plasmid, and the plasmid produces one protein. I mean, it's a recombinant plasmid, it has gene, and you can express it, or before expression, you can multiply the transformant, and you will get, say, for example, 10,000 transformants by amplification. Means you're just culturing the transformant. And then after you have done this, you would have added the inducer. Say, for example, you added IPTG to induce the transcription from lac operon. Assume that the promoter that we have here is lac promoter. And if in each cell, if there is 100 molecules produced, then you will have, say, 100 into these many protein molecules, right? That is one possibility. That's a good way of doing things. That is typically what people do. You have a transformant, you amplify them, then you will induce using an inducer. Here I took IPTG, and then you have a large quantity of proteins. They will usually have more than this. And then you will purify and characterize it. What if the protein is toxic? That means one or two proteins produced will kill the cell. Okay, so if it is toxic, how can we assume now we're talking about the toxic protein? A gene is produced. If the gene, even basal level of expression would kill the cell, then how can we get 10,000 cells to start with? Even if we get 10,000 cells, then we will probably have one protein per cell, say, theoretically. And then we are left with very small number of proteins. In those cases, or where some of the proteins are very small, some of the genes are very small, and those small ones are, if they are produced, they're usually degraded by the cellular proteases. And then too, we cannot get high concentration of those proteins, and then further characterization will become very difficult. Excuse me. So in those situations, we can insert it into a protein like this, make translational fusions like this. So as a result, what may happen? Now the protein that is produced, I'm putting the toxic one here. And when it is produced, it may not have as much toxicity because it is fused with some other protein. And there are ways how you can get rid of it also. I mean, finally, you actually want only this part of the protein because that is the toxic part and you want to characterize it. As you will know, you might have studied or you will study, there are ways how you can engineer a site for cleavage here. And then you can cleave the protein exactly in there. And then you can isolate the protein that you're talking about or protein of interest, and then go for characterization. So that is another use of how fusion proteins may help. That's the same case with the small peptides or small proteins also. I hope you understood that. This is based on lambda GT11. You can do it. Plasmids also are there where you can produce those. But we will see another example, which is called as phage display. Okay, sorry. Well, that will be the last, and I hope I will leave you with that. And then I'll repeat that again in the next class. So I have a situation where, for example, like we have already discussed about gene library. I want to find out which all proteins actually bind to, which all proteins bind to X, okay? A molecule or a protein called X. I have a gene library. I want to find out proteins that bind to, okay, bind to X. So you can do it also. I mean, you can have variations of this experiment as well, but right now we'll just go with it. So I would have made a phage display. What is this phage display? In the sense, as I discussed, there are different phages. On the surface of them, you have several proteins, like we have seen for M13, for example. So you take the gene of that protein, the phage, I will just put it as capsid. Okay. You recombine, you make a translational fusion of the capsid or one of the non-essential phage surface protein, the gene, and then you make a translational fusion with the gene library. Okay, when you're making gene library, we used in the previous one, we took, I'll just go there so it's easy for you to recollect. We took the the genomic DNA, processed it, and we added SAU3A and all those things. Here our function is to construct gene library, and hopefully we go for sequencing, chromosome walking, and so on, which we will discuss later. The same thing until this step we could have done, and then we could clone it into a vector that has a cloning site exactly after a capsid gene. Okay, this is the cloning site. In one case, you may have gene A cloned. In another case, you may have gene B cloned. Like that, you will form a library of recombinants capsid, and in other case, you may have gene C cloned, right? You're essentially making a, you're essentially making a gene library using this. The everything else is present. You have the promoter, there is capsid gene and gene of A, B, and so on. Here it is represented by this. So, okay, let me get through this. When this forms a protein, it will form a capsid protein, and which is translationally fused with the gene, with the one of the genes from the gene library. And if it forms a phage, if this may form a phage to which you may have the capsid might be embedded in the membrane or so like that, and to which you may have the display of, because these two are translationally fused. So this is from gene. This is from the gene library. Assume this is a protein A. You may have several of those. In some case, you may have protein A. In some case, you may have protein B. And that is where you have constructed a phage library, okay, with translationally fused phage library. And this is what we are trying to do, is use it for a purpose, right? Now, in one case, it is gene A. In another case, it could be gene B or protein B, C, D, or E, and so on. As I described in the objective of the experiment is to find all those protein and all those genes which encode proteins that bind to protein X. So I'll try to use this here. So we take a plate or solid phase, stationary phase, coated with protein X. These are the phages that we have made with translational fusion from the gene library. One of it may be having gene A, gene B, gene C, gene D, and gene, say, EF. And then we treat all these phages, we allow the phages to bind to the stationary phases. Only those that have affinity for it will bind, and the rest of them will be washed away. And then we can isolate the phage, isolate the DNA or the vector that we have, and then sequence, or you can further clone it and figure out which one, what is the gene name, what is the gene sequence, and then you can figure out what it would encode, right? Our objective is, remember, I have given a broad objective. I don't know anything about a vector. I'll just describe that once more. I gave you an organism, or say fungi, and we have indications, or we want to just explore. It produces so many genes. It has so many genes. It produces so many proteins. I want to find out which of those proteins bind to protein X or ligand X. How can I do that directly? We don't have anything. Then we would have isolated the genomic DNA. From the genomic DNA, we would have made gene library or genomic library. And when we are making the vector that is used is using translational fusions, because we are trying to employ the principle of phage display. That means the protein should be displayed on top of the phage. Okay, if this is phage, this is capsid protein, and this is the gene of interest probably displayed. If you want to do that, we have to make the translational fusion of the library, or in one case gene A, one case gene B, and so on. So this is the cloning site, and here is a capsid gene. So when we make like this, we will get multiple of those. One may be having gene A, one may be having gene B, and so on, C, D, E. Then we would have infected and produced several phages. And those phages, we will, because now the phage is produced, phage will have the capsid protein, and on top of it, you will have the gene of interest, either A, B, C, D, or so. Different phages will have different of those. We will treat a stationary phase that has immobilized protein X and see which all, and then we wash away all the phages that do not interact, and then be able to isolate the phage that binds to protein X. And that's how we get to find out which of these genes and those encode proteins that bind or have high affinity for protein X or ligand X. Okay. If you have any questions, I will take. It's already 5. So if you have any questions regarding CIA, did I already? Okay. Firstly, anybody has any questions on what we have discussed today? We discussed about cosmid advantages, and the primary reason is for adding cos site is to get the DNA packaged. And why do we want to get it packaged? Because we want the DNA to be delivered into the host cell much more actively using the injection system or something of lambda phage. So we have cos site. So and if we use cos site, because we are adding on cosmid, then the size that can be cloned can be larger, something like 20 kb or so. And then we have discussed about single-stranded vectors, which are usually mostly about M13 phages. They are single-stranded vectors, and they produce a replicative form. They become double-stranded, which is easy to isolate as plasmid isolation, and then we could do manipulation and put them back or transform back into a host, and then get single-stranded DNA. Single-stranded DNA are important for sequencing in the olden days. Nowadays, the sequencing technology is revolutionized big time, and also used for probe in case of southern blotting. It can also be used in mutagenesis, site-directed mutagenesis, and so on. And then we discussed about expression vectors. In that, expression vectors need to have a promoter and a ribosome binding site so that the protein can be produced. We also have transcriptional fusions, where we have a promoter of one gene, another element, and we fuse it with a gene of interest, then it is a transcriptional fusion. We can also have a gene from the host and a gene cloned downstream of it with a gap. There should not be a terminator between them. Okay, that is one of the important things. If you have terminator between A, B, and X, then only B is produced and X cannot be produced. Then we discussed about translational fusion, in which we have the end product will have a part of the host protein and a part of the gene of interest that we have cloned. One important aspect is that you should ensure that the reading frame is continuous without any alterations. I hope you remember frame shift mutations. Please go through it, and if you have any questions, we can discuss again. And then we discussed about one case scenario of what is the use of producing these fusion proteins, especially for those toxic proteins or small proteins or difficult to clone proteins. They use translational fusions. When you have here, we will get to those, some of the examples. You can include a cleavage site so that after the fusion protein is produced, you can treat with the protease that cleaves specifically in this site, and then we can get the protein of our interest. And then we discussed about phage display. Phage display is on to display the protein fragment or gene, the protein on top of a phage. And if you want to do that, we should have made translational fusions of the gene with the capsid, because capsid makes the or the cell surface or the phage surface proteins. It not necessarily have to be a capsid. And in that case, it will produce a translational fusion protein like this, and which will then be also present on the protein phage surface like this displayed. So you call it as phage display. Okay, I'll stop here. If you have any questions, anybody has any questions?
Sir, this is with regards to the phage display.
Little louder, please.
Sir, am I audible?
Yes.
Like, is it better?
Yes.
Okay.
Yes.
So this is with regards to the phage display. So you mentioned that the fused proteins would be expressed on the surface of the phage particle because it is going to be fused with the caspid protein.
Yes.
But how do we know for sure that it will be expressed on the outside, not the cytoplasmic side, like the inner
Yes.
part of the phage?
I'm sure many scientists have tried many different proteins. They must have failed in many things. So when I say they should have done with it, these are already known proteins. That the addition of some part of it as a protein like this did not affect the function of this protein, the capsid protein, to the phage. Okay, we didn't know those things. We only tried many of them, and then we are successful with some. We cannot randomly take any capsid and just put these make translational fusions like this and expect it to function properly. Yes. Did I answer your question?
Yes, sir. Thank you, sir.
Okay. Is there any other question?
Regarding the exam pattern, sir.
Why so low? So exam pattern will have 3 questions. I already told, or I didn't tell you people. This will have 5 bits, and each one is 1 mark. Okay, just define kind of things. So that is 5 marks. And this one is 1 question for 5 marks. And this is 1 10-mark question. Okay, so question 3 will be analytical. These are simple remembering level. So you should know, if I told you to read by yourselves, you should have read them carefully. Okay, so these are mostly remembering and you showing motivation by finding the answers yourselves, or majority of them I think I have discussed. So please ensure whatever is covered in the syllabus until last Saturday, that is there in the syllabus. Remembering, and this will be an analytical question. I have discussed, I have gave you some assignments as well, or videos to watch, and you should be able to analyze those, right? And then the 5-mark question will be something probably explanatory and so on. Okay.
Yes, sir. Thank you, sir.
Yes. Anything else? Hello.
Nothing, sir.
Nothing else, sir.
Okay then. So yeah, you have to be right on. You have to be 5 minutes early. You'll have to put your video on, and the exam will be recorded. So ensure your surroundings and you are professional. Okay? Is that fine, everybody?
Yes,
Please
sir.
tell others who may not have been here or who are not watching, please tell them to do that as well.